Today's topic for discussion is a practical protocol of plant pathology. Let us discuss the practical of damping of disease of vegetables. It is one of the very common disease in Marathwada as well as throughout India, and it is a cosmopolitan disease throughout the world. Damping off is very common because every country grows the vegetables. And what we have to do during this practical, let us see. Next, this what is the requirement for the practical? Is the requirement is infected seedling. Again, it will be a that museum specimen you have to use. Museum specimen of infected seedling. Okay, slide, cover slip, watch glass, blade, cotton blue, lactophenol, and blotting paper, needles, microscope. All these are the common things for every practical. We need it, and it has to be it is to be available in the laboratory, otherwise it is difficult to do practical. Examine infected seedling and note the morphological abnormalities found. Okay, this is one of the procedure. Or one more method we can use for the practical purpose. You can isolate the pathogenic organism from infected plant part. This is a scientific method. But in BSc third year syllabus, as per UGC norms, we have to observe the specimen and you have to write the all details of the symptoms and the causal organism. And if you decide to isolate the pathogen, what you have to do? After survey, you collect infected seedlings from the field in a sterile polythene bag, and that has to be brought to the laboratory for further work, and you isolate the pathogen from the infected plant material. How to isolate the pathogenic organism? Take it in the laboratory, that infected material, then rinse the material with distilled water, then again you rinse with 0.01% HgCl2 solution, again rinse 2 to 3 times with sterile distilled water, and then take it into the laminar airflow. Prepare a medium, mostly PDA medium is used, and potato dextrose agar medium, amended plates, you make it ready. And plates should be sterile before adding medium into the plate. The plates, petri plates are generally sterilized in autoclave oven. It is a dry sterilization method. At 120 degrees Celsius temperature, you have to keep the plate for up to 24 hours. Then it has to be thought that the plates are sterile. Okay. And in sterile condition, in the laminar airflow air chamber, the media, sterile media, we have to add into the sterile petri dishes in sterile condition, and when after 5 to 10 minutes the medium becomes solid into the plate, and then you have to take a seedling which we have kept there by washing with water and HgCl2 solution. Cut a small piece of the seedling and keep it on the media amended plate. Close the plate and keep it at the room temperature. This phenomenon, it is known as incubation. We have inoculated the infected material into the plate, and then we have incubated. After incubation, observe the plate every day, and for its further developments, you will get the small thread like growth from the infected plant part, and make the slide and observe the pathogenic organism. What happens, several fungi grow on that infected material, even the media is there. To avoid this, again re-inoculate several times by removing a single thread of the pathogenic organism, and purify the culture by the method of single hyphal thread inoculation method. Okay, remove a single thread, inoculate in a newly prepared PDA amended plate like this. And pure culture, you can observe under microscope. Take a drop of glycerin. This mycelium, it is a hyaline. Therefore, you have here we have to use. You can use the cotton blue. Okay, because the mycelial threads are hyaline, and in cotton blue they can be seen very clearly. And use the cotton blue and observe under microscope and write down all the details. This is one more method. Okay. Otherwise, you have to observe that infected seedling in the museum specimen, from the museum specimen, and then you have to write the details and write down about the the pathogenic organism. Next, this is the classification of the pathogen which is responsible to cause the damping of disease of vegetables. The pathogenic organism is Pythium dibarianum. There are several species of Pythium which is a soil borne and which causes the damping of disease of several types of vegetables. Okay, next. In the mycelium, what exactly we have to observe? You have to observe a non-septate mycelium or sinusotic hyphae, and sporangia will be there. Some of the straight hyphae, they act as a sporangiophore, and at the tip of the sporangiophore, sporangium is there, which is mostly lemon shaped or oval structure. In the section or in the culture, which is grown pure in the pure culture, oospores are also seen. And the sporangia may be terminal or intercalary. You have to write the size of the sporangium, 115 to 26 mu in size, oval or prominent protuberance at the tip. Okay. And the oospores are smooth walled, which are mostly 12 to 20 mu in size. These are the details of the pathogenic organism about the mycelium that you have to write by observing the slide of pure culture which you have isolated in the laboratory. Next, the pathogenic organism, which type of symptoms are caused? We know the theory that this disease occurs in two stages, that is pre-emergence damping off and post-emergence damping off. When the seed comes, after sowing, what happens when the seed comes in contact with the pathogen below the soil surface, that get infected and that seed get rotted because of the infection of the pathogenic organism Pythium afanidermatum or Pythium dibarianum. What happens? The seed get rotted and that do not germinate and do not comes out of soil surface. Such seeds, that phenomenon is known as pre-emergence damping of symptoms. Okay, that means rotting of seed takes place below soil surface. We never see the seedling, it do not germinate. Okay, that is pre-emergence damping of symptoms. Okay, and when the seedling germinate, that comes out of the soil surface, at that stage the pathogenic organism infect the seedling, and the girdle of the stem just above the soil surface shows water soaked spot, and gradually that water soaked spot that extend around the seedling, and entire seedling get rotted and fell off on the ground. This stage of the pathogenic organism which causes such type of symptoms after emergence of the seedling, after growth of the seedling, after the seedling is visible out of the surface, that stage is post-emergence damping of disease symptoms. Okay, this you have to remember and this you have to write down. Let us see next. This is the seedling showing damping of disease of vegetables. Okay, next. Thank you very much. Save trees, save earth. This is all about damping of disease of vegetables. At present, because of the lockdown, we are teaching online. Hence, practical is difficult to do in the laboratories. Hence, we have started this method of demonstration of practical for BSc third year students. Thank you very much. Save trees, save earth.